which tells about the when the gene gets expressed. Okay, when all genes get expressed. Gene expression means transcription and translation, and that in one line, if I say, it means when a cell has to undergo transcription and translation for the process or for making proteins. When a cell has to make protein, that information is actually uh, decoded by the DNAs in the form of transcription followed by translation, and we get the protein what the cell needs for making of something, or for, for some particular function, for whatever reasons. For suppose a formation of an enzyme, it needs a particular protein to make enzyme, because that enzyme is needed for the digestion or the breakdown of further something. So in that cases, when it is needed only, it has to be produced. So that kind of gene expression, the regulation of the genes, which further helps in the transcription followed by translation, is known as gene expression. Okay, so in this topic we are going to learn, I was telling you regarding this lac operon model. This lac operon model will help us to understand how it actually works in our body, also in all the bodies, about when the cells or when the DNA transcribes into mRNA, further it translates into a protein. Okay, only in the, since all this process is a lot of energy consuming process, so it is done only whenever it is needed. Okay, whenever it is needed. So the point is where this small process, I mean not for small process, but this process can be understood by this lac operon model. We will start this today. Okay.
However, lactose needs to be hydrolyzed or digested before it is used, and hence the bacterium prefers to use glucose when it can. But in the absence of glucose and presence of lactose, E. coli synthesizes an enzyme, beta-galactosidase, to catalyze the hydrolysis of lactose into galactose and glucose. However, in the absence of lactose, the bacteria will not synthesize the enzyme beta-galactosidase. This shows that gene expression depends on the metabolic.
Okay, so the point is, the first thing comes here is, we have taken this example, the lac operon model explains in prokaryote, that is in bacteria, they're explaining E. coli bacteria. So here the organism uses glucose as its major resource or major source for getting energy. Okay, so every time in the body, if they have glucose, they don't need to have to go undergo these processes. Okay, because glucose is a monosaccharide, it does not need any breakdown or digestion. It can be directly used under or in the process of respiration, as we have learned, and it will yield energy to the E. coli bacteria. So that is clear. So if there is see the conditions when it will start. Okay, what makes it trigger this E. coli, this lac operon model, when it becomes active? The only reason it becomes active is when there is no glucose. Okay, glucose is the component which is needed to enter into the respiration, and then which will yield energy. Okay, as it needs energy for all its physiological processes. So if there is glucose, it will not undergo any uh, further process for making that enzyme. But what happens sometimes, what happens inside the cell of E. coli, they don't have glucose at all, but they have lactose. If there is lactose present, lactose is a disaccharide. Okay, it's a disaccharide, so it needs to be broken down into monosaccharide. Then only it can be used in the like the respiratory processes to yield energy, that all those glycolysis and all, right? So it has to be brought into monosaccharide. For breaking it down into a monosaccharide, a disaccharide has it needs an enzyme. Okay, this enzyme which E. coli uses to break down lactose into glucose is beta galactosidase. Beta galactosidase. This enzyme is needed for the breakdown. Okay, now, so the first condition for this gene getting excited, for the gene expression, is that the E. coli bacteria should not have glucose in the cell and should have only lactose. Okay, so the first condition is that it should have only lactose. So if it has only lactose, what will happen? It definitely needs the enzyme, so it will get expressed, the genes will get expressed, and it will create mRNA, that is transcription followed by translation. Okay, so the first condition is very
physiological
important when
or
we start.
environmental conditions that exist in the cell. Moreover, gene regulation allows the cell to control its structure and function. This is the basis of cellular differentiation, morphogenesis, and adaptability of any organism. In fact, the development and differentiation of an embryo into an adult is also the result of coordinated regulation of expression in several sets of genes. In prokaryotes, the predominant site of
Now we have not entered
gene regulation is the
not
initiation of
entered yet in
transcription.
the process. Huh? Okay, it is telling.
In the transcription unit,
Okay, it tells that what they've shown you just now, the image of right from the infant after the birth, or from the birth time till the conception, from the conception time till the we are going to learn through lac operon is an example only. This shows inside our body every time this process, gene regulation, is happening, and how it is happening is we are going to learn from lac operon. And they're saying gene expression is such an important, such a vital activity inside our body, because it is because of these gene expression only we or the infant or any activity inside our body happening, any, because every time our body needs proteins which needs for the, like for making of the cells, for making of hormones, enzymes, for anything in our body, like repairing of the cells, damaged cells. Every time the RBCs after 120 days has to be made, synthesized. So any cell when we say has to be made, it needs proteins, hormones, enzymes, all are made up of proteins. So when they have to be made, so you can understand in what quantity and how much this gene expression is happening inside our body. So for all these processes we need protein, and protein is formed, as we have learned right now, from the transcription further followed by translation. And and see, there is nothing extra happening inside our body, so everything is very much controlled. Okay, and this control, for that, how it is getting controlled and how we are able to do this with the source of energy what we have, all this thing we are going to learn. So gene expression is a very important factor which happens, as I said, uh, right from the conceptual time in our body till we die, because some or other cell is always needed to be formed and reformed. uh, or proteins are always synthesized. Okay, so the process of lack operant model we are going to learn. This is the this what they explained to you now is why this or why we say that gene expression is very important.
The interaction with accessory or regulatory proteins regulates the ability of RNA polymerase to recognize and bind to a promoter, that is, the start sites. When regulatory proteins act positively, they are called activators. And when they act negatively, they are called repressors. The accessibility of promoter region.
Okay, in one line, if I will say this process, which is going to come to you only in an animation where you can easily understand. In one line, if I will tell you, is the process of transcription comprises of three things. Okay, the transcriptional unit comprises of three things, that is initiator, then structural gene, or and a promoter, structural gene, and followed by terminator. If in the promoter, or the what you call it as initiation point, the code which defines it, that where the RNA polymerase enzyme has to come and sit. Okay, in that place, if something else is sitting, okay, if on that code something else is already has occupied the place, what will happen? RNA polymerase enzyme will not be able to bind itself in the promoter area, in the initiating part, the code where it says to start, okay, or from where it has the information to begin. Okay, on that codes, if and something is already there, okay, it will not allow the further process to happen, because the promoter area itself is not allowing. Okay, because something else is over there. So that is known as repressors. Okay, so repressors are again proteins only. They are also made by transcription and translation. Every time it is made, and they just like a lock, you can say, it will lock, it will remain binded on the promoter area as a lock. They will remain there, and it will not allow the RNA polymerase enzyme to come and sit on that code. So what happens if this repressors are always available on that part, and it will not allow the initiation process. Okay, so the presence of this repressors will not allow the process to proceed. So whenever the body needs the enzyme, the repressors move out. The repressors move out. Then only the promoter area, the initiation processes happens with the RNA polymerase sitting on the initiation point. Okay, so the repressors again are proteins which are again synthesized every time, and it binds the area of promoter. Okay, and the removal of that repressors, okay, will allow the transcription to happen. And when the repressors will be moved, only when in we are talking about lac operant model, only when it gets removed repressors. Again, repressors are protein. They are removed only when in the cell there is no glucose, only and only lactose is present. That is the only reason when this repressors move, and allows uh, the process of transcription to proceed. Okay.
Regions of the DNA in many cases is regulated by the interaction of proteins with sequences called operators. The operator region lies adjacent to the promoter. In most cases, a repressor protein binds to the operator sequence, due to which the operator is inactivated, and hence transcription does not take place. But when the repressor protein is removed, it activates the operator, and hence transcription occurs. The regulation of gene expression can be studied by the operon hypothesis. Let us now understand genetic
As you heard, it
regulation
is
with the example of
hypothesis.
lactose or lac operon in E. coli. The lac operon was described in detail by two people, a geneticist, François Jacob, and a biochemist, Jacques Monod. They were also the first to explain a transcriptionally regulated system. The bacterium E. coli uses either glucose or lactose as a source of energy. Depending on the source of energy available, there are four possible situations that exist in the bacterium, as shown in the table. Before we understand the lac operon in detail, let us understand what an operon is. An operon consists of a structural gene which codes for several proteins and is regulated by a common promoter and regulatory genes. Each operon has its
Okay, this is what you call it. This is a prokaryote. That's why it is polycistronic. Okay, that's why the structural gene, as you can see, the structural gene, pen is missing. The structural gene you see here is with a green color, red color, purple color. It is written there, and that is different types of proteins are. This is a polycistronic type. Okay, polycistronic. That's why you can see this. You have to learn this operon model with these names. Huh? We have to learn all these names. You have to learn the structure. What is what? Okay, so that we will learn right now. But I'm saying this is the this points are over. Polycistrons means this is a prokaryote. It will not carry one in one. This is one type of protein. It has the information of second type of protein, third type of protein, like this. It will go on. Okay, that is a structure. What it is written here, structural gene. Okay, the structural gene carries in eukaryotes only the information for one type of protein, while the structural gene in a prokaryote are actually polycistronic. Okay, and what is this? This is DNA. Okay, fine, and this is I mean the process what is happening. Fine.
specific operator and specific repressor. For example, the lac operator is present only in the lac operon and it interacts specifically with the lac repressor. The lac operon consists of one regulatory gene, or I gene, and three structural genes, lac Z, lac Y, and lac A. The I gene codes for the repressor. The structural genes code for enzymes required for the metabolism of lactose. The lac Z gene codes for beta-galactosidase, which hydrolyzes lactose into galactose and glucose. The lac Y gene codes for permease, which increases the permeability of the cell to lactose, while the lac A gene codes for transacetylase, whose function is unknown.
So when you this one, okay, this lac I is meant for the later on it will, whenever it will translate into protein, it will make the repressor molecules which comes and binds on the operator area. Okay, now lac Z is for the this, as I said, when they are saying enzyme, it will not make directly enzyme. It has the code for synthesizing, making the mRNA followed by proteins, which will make enzyme, which will be helping the lactose molecule to break down into glucose and galactose. Okay, so that is lac Z. Likewise, this lac Y and lac, these are the three different proteins which are produced. This permeases, this one lac Y is the code in the on the structural gene, which will decode into or synthesize the protein, which will make the enzyme called as permease, which will make the cell wall of the membranes of the E. coli permeable for the entry of the lactose. Okay, same. I'm telling you, same structural gene carrying more than one protein information. Okay, that's why polycystron. While this lack this okay, sorry, this one is translase is not yet understood what is a function of this this protein. It's not yet discovered. Okay, so this is the model of lac operon. Till now we are going to enter only, as I said, this lac operon model. uh, the first three four things in this, you have the structure you have to understand. This is the E. coli bacteria. This is just they have made it for to make you understand about the inside this, on this DNA it is happening. Okay, all these things. Now this is what they have made it in a, in this form they are explaining you. Okay, now that is one thing. Then this is for repressor molecule. Okay, this is the DNA, of course, and this is a repressor molecule. This is the message or information which carries for forming of the repressor molecule. Again, as I said, it is made up of protein. Okay, when all this will be only, see, whenever lac I gets transcribed and translation happens, it will form the repressor molecule and it will bind the area of promoter, and further no, nothing happens. This nothing will happen. Okay, for making of and if suppose, and when this will happen, what is the condition this will happen? That is when all the repressor molecule is synthesized or made, is when the E. coli has got glucose enough for its source of energy. As you know, when glucose is there for the source of energy, and it does not need the enzyme to be made to digest or to break down the lactose till that time. Okay, till the time the E. coli is having glucose, the repressor molecule always will be active, and this lac I will be transcribed and will form translation and form the protein and will make the repressor and will bind the promoter area till it has got glucose. The point, even when the glucose is absent and even lactose is absent, even that time also lac I only will be produced and it will bind the repressor, will come and bind the promoter, this operator part. Okay, that means when glucose is there, that time also repressor molecule remains there. When lactose is absent in the cell, that time also it remains binded over there. Now this repressor moves only when there is no glucose at all in the cell and only lactose is present. That is the time. Remember, the condition is only lactose. That triggers. Okay, that's why the model's name is lac operon. Okay, so it triggers and then the repressor molecule, this is not, it's not activated. Okay, at that time the repressor moves out and this lac Z, because for digesting or breaking down the lactose disaccharide molecule, it needs beta galactosidase enzyme. Beta galactosidase enzyme is needed, which is synthesized, which is done with the help of the protein. For making of this enzyme is brought by lac Z. That is the time the repressor moves down and the process begins.
The switching on and off of the operon is controlled by lactose and is hence the inducer. Since lactose needs a small amount of permease enzyme to enter the cell, a very low level expression of the lac operon is continuously required. The lac operon functions in the following manner. The repressor protein is continuously synthesized from the I gene. The repressor in its active state binds to the operator sequence, thus preventing RNA polymerase from binding to the promoter site and transcribing the operon. In the presence of the inducer lactose or allolactose, the repressor is inactivated and transcription proceeds. Thus, regulation of the lac operon is an example of regulation of enzyme synthesis by its substrate, that is, synthesis of the enzyme beta-galactosidase by its substrate lactose. This regulation of the lac operon by the repressor is called negative regulation. In this way, the lac operon demonstrates how gene expression is regulated in an organism. In the past, biological researchers could study only 1 or 2 genes at
नहीं हो नहीं रहा बोल रही हूँ मैं to tell about what is human genome project. Okay, to write about human genome, because it's a many years it took and many scientists, and then they have produced the whole human genome project, which is so difficult to pen down on a book. They say if we are going to write it like in our what informations a human is carrying inside the genes, that is human genome project. If you are going to pen it down in the pen paper, it will take around a book or a copy which is having 1,000 pages. Okay, and each page with 1,000 letters to be written. That much information our body carries in the form of human genome project, and its silent features that what all things, it's like complete uh, knowledge about the human genome project we are going to listen, okay, after this. Okay, HGP, human genome project, this comes of 3
taught
marks.
the entire DNA sequence of a single human cell in books, we would have 3,300 books, each with 1,000 pages, where every page contains 1,000 letters. Yes,